Good afternoon, everyone. It's our great honor to welcome you to the third lecture series, Presidential Leaders in Science, the Presidential Lecture Series, a distinguished platform where visionary scientists and thought leaders shared groundbreaking insights that shape the future of research and innovation. Curated and hosted by our president, Professor Dr. Metin Sitti, this series highlights pioneers at the forefront of their fields, fostering intellectual dialogue and inspiring our academic community. Today's guest of honor is practically a VIP around here, as this isn't his first time with us. I guess it's going to be his third time with us. So we're so excited to welcome him back to Koç University, to our campus. To introduce him, I would now like to hand over to our president, Professor Dr. Metin Sitti. Thank you.
Hi, everyone. I'm very delighted to introduce our today's speaker. Before that, I welcome you all, and also our chair of Board of Trustees, Professor Nur Yalman, is also with us. As Hande mentioned, Kamil Hoca is very eminent MR scientist, and he's also member of our Board of Trustees. And very briefly, his story starts maybe back in Izmir, and then Robert College. And after Robert College, he went to US directly. In those times, it was not common. Now it became too common, unfortunately. So he went to Columbia University, and also Nihat Berker, at that time, his friend, went to MIT. So they were two well-known physicists from Turkey. And in Colombia, he studied bachelor's and also did PhD there in physics and then also chemical physics, if I remember. And then he did a 1-year company software job, and then in Bell Labs he was there for some years. Then he became a faculty in Colombia, but then Minnesota, I think, gave him a great offer to go there as a faculty, and since then he has been in Minnesota. So I know Kamil Hoca through a lot of network in MR imaging, especially fMRI, when you talk about instrumentation, physics, and methods. He is so well known. I heard him from my ETH colleagues many times, but it was a great pleasure to meet him through our Board of Trustees and Koç University. So in Minnesota, he is leading a very important center on magnetic resonance research. I heard more than 200 people now, with a lot of faculty and students and researchers involved in such amazing center, and the research is especially focused towards understanding brain function, connectivity, and also anatomy. So he's a faculty in medicine, so from physics to medical school in both radiology, neuroscience, and also I think medicine, let's say, as affiliations. And but he's again a physicist, and today he'll talk about his research activities in recent years. He's very well recognized, and I hope he's on the pipeline for Nobel Prize, if there is any Nobel Prize for fMRI one day. And so basically many awards, but important ones to mention is one is National Academy of Medicine. He's a member there, and also he received our VP Coach Science Prize. That was some time ago, and also many other recognitions that he received. But it's great to have him here. So as I mentioned, he comes frequently for the board meetings, and I really highly encourage all of you to listen and also then interact with him for possible collaborations, even consulting to him for research and many other things in life. He's a great sailor and maybe many other skills, very active in sports. So he's very slim, and all these things are important besides science. So again, I'm very happy to have him here today as our presidential seminar speaker, and let's welcome him from Minnesota.
Okay, it's a great pleasure to be back here, I say, because as Metin has mentioned, this is my third lecture in Koç. First time I came to campus in person, that was 2018, and then the students invited me to a student forum, and of course, when Koç students invite me, I do not say no. And I gave a lecture online. That was an online lecture, and now this time again. So, in fact, as a result, some of you may have heard me talk here before, and for those of you who heard me talk here before, some parts of this lecture will be a repetition, if you like. But as we go on, then I will get to new things as well. And for those of you who have not seen my previous lectures, I hope the whole lecture will be interesting. It's unfortunately a long lecture, and I was given the freedom to have a long lecture, and then later on and earlier today, when I was talking to the faculty members and we were talking about some work that was not included in my lecture, and there was interest, oh yeah, that would be nice to hear as well. So the lecture got longer between this morning and the actual lecture. So it's a difficult lecture in some ways because of the fact that it's really a multidisciplinary topic. It involves a lot of physical sciences and biological sciences. And so to emphasize that multidisciplinary topic, I wanted to start with maybe introducing you a little bit about my group. Let's see. So this is a picture taken for the Center of Magnetic Resonance Research a couple of years ago. You can see there are quite a lot of people, and they come from various different disciplines. We have physicists, engineers, mathematicians, neuroscientists, who actually work very collaboratively in this center towards developing new techniques to study the brain. And in particular, I'd like to call attention to our engineering group. And I know that engineering is a rather large faculty here, and engineering is also very important to us. And some of them are actually from Turkey as well, and they are really the ground upon which we build most of our research. And this engineering group, of course, evolves. People move on to other things, and so some other people who were not in that picture. And I also want to emphasize this particular group, and they are mathematicians, and it's a very important topic for us in terms of signal processing and in terms of image reconstruction. And in CMRR, then the research has many components. We, for example, study animal models. We, of course, study human, and there's a large emphasis on human instrumentation and methods. And in this particular case, of course, we have physics and electrical engineering, signal processing and image analysis play a very important role. I made it intentionally as kind of like a inverted pyramid because all these other studies, applications, biological applications, really are based on the methodological developments that we undertake. And that is why I wanted to emphasize the role that engineers play in the center. And our main drive in terms of this instrumentation and methods is the really development of tools. So we want to develop tools to be able to advance the boundaries of biological information. Now, many of you, including me, would say, well, that's self-evident, obviously. To advance knowledge, you need new tools. But it's amazing that a lot of scientists, important scientists, somehow feel the need to impress upon the world that this is an important thing, to be able to develop tools. And you can find a lot of quotations. One of my favorites, in fact, comes from this person, Leroy Hood. I had the pleasure of meeting him some years ago. And then before I met him, I was looking up, okay, what has Leroy Hood done? And I found out that he had played a very important role in instrumentation of DNA analysis. And I found in his biography that he credits his PhD advisor who told him, if you want to practice biology, do it on the leading edge. And if you want to be on the leading edge, invent new tools for deciphering biological information. And this is something that we follow. It's a philosophy that we follow in my lab. And I have to say, I mean, nobody really taught me that per se, but Metin has mentioned that I worked in Bell Laboratories, and that was the culture of Bell Laboratories. Always push the tool frontier to get new information about biology and many other things. And this particular aspect of tool development was also recognized by NIH at some point. I say at some point because for quite a long time it was really hard to get funding from NIH for tool development. But then there was the realization that we really don't know very much about the human brain, and human brain diseases are not has not evolved, and we need to do something about that. And there were a couple of projects that came into existence. One of them is this Human Connectome project that I was very much involved in, and the other one is a initiative that was launched during the Obama administration, and it's so-called Brain Initiative. And these projects are in fact very much focused on tool development, and very interestingly and very satisfactorily for us was that we have been doing that already for quite a long time before these projects were announced. And so as a result, we were really in a position to take advantage of these projects and use them to advance our research in our laboratory. So the tool development that we are interested in mainly is magnetic resonance. And what we are known for is really pushing so-called high fields for human imaging. So, for example, this is a picture of a magnet which operates at 4 Tesla that we got from Siemens in 1990. It looks like a very primitive object. Maybe Siemens was not aware of color photography at the time, but it's a very advanced magnet. And at a time when magnetic fields in hospitals were at 1.5 Tesla, then we developed a 7 Tesla. Again, this is the very first time we went to 7 Tesla, and I will tell you that there's quite a lot of physics that we had to understand and develop instrumentation. And nowadays we are working at 10.5 Tesla. This is the 10.5 Tesla magnet being installed in our laboratory. As you can see, it's a very large lab system. But as a result of these initiatives, we have a lot of magnets, and the old magnets, which are very historical magnets, do not disappear in our center. They become instead garden statues. So we have a courtyard, and this is the 4 Tesla magnet that you saw, and this is the very first 7 Tesla magnet, and they are now garden statues in our courtyard. Now, the rationale for going to these high magnetic fields, as I said, at a time when clinical MR scanners operated at relatively low magnetic fields, was based on functional imaging. And functional imaging came into existence when my colleague from Bell Labs, Seiji Ogawa, discovered this phenomena. So if he was studying rat brain, so this is a rat brain, and you can see that there are these dark lines in these rat brains. And when he then manipulated the metabolism of the rat brain. For example, you can give glucose, or you take away glucose using insulin, and you can actually manipulate those lines and they disappear. For example, in this case, you reduce the blood glucose level, so brain does not have a carbon source, and the lines disappear. And then you give back some carbon source, and the lines come back again. So he was very correctly identified them as a result of deoxyhemoglobin in those blood vessels. Those are blood vessels. Now, this is not functional imaging, but later we use that phenomena to introduce the ability to map brain activity using that particular phenomena called, he called it BOLD, blood oxygen level dependent mechanism. And this is the paper in 1992 that introduced functional imaging. And at the same time, there was an independent effort from MGH, and this is so as a result, this is one of the 2 papers that introduced it. And there's a picture of some of the authors. Is you can see me, Seiji, two of my postdocs now, they run their own centers and at a meeting in 2019. And this is the first image that we published showing activity in the brain. And the subject was sitting in the magnet. It was dark in the magnet. And we had lights come on at some point and lights came off, lights came back on, then came off. And then when you look at images, so we are collecting one image after another. And if you look at images, the intensity of these images, for example, you look at this region, nothing happens. But you look at this particular region, you see signal changes tracking this lights coming on and off. And of course, you can generate an image based on this. You can make a, for example, subtraction between these areas and that areas, and you can put that as a color map. And as you can see here, and that's the activity of the brain. And of course, this is visual stimulation. We use visual stimulation because it's very well understood where the brain should react to it, and you can see this. So that was a very exciting development, and now functional imaging is the most commonly used technique for studying the human brain. Now, you need to do that, a magnet, and we use this 4 Tesla magnet, which is now a garden statue. And the other thing you need, of course, is water molecules. Our body is full of them. We are looking at nuclear spins of these hydrogen atoms. They are microscopic atomic magnets, if you like. And we are monitoring the signals coming from that. These magnets interacting with this big magnet. And then we actually need another magnet, and that other magnet is sequestered in the red blood cells, and it is hemoglobin. So hemoglobin is, this is the hemoglobin molecule, is strongly magnetic in its deoxygenated form, but not in its oxygenated form. Now you have to combine that information with another information that comes from Cambridge University in 1893. And Roy and Sherrington, who won the Nobel Prize for this particular discovery. And what they discovered was that blood flow in the brain is coupled to neuronal activity. So when neurons are activated, blood flow increases. And there was another very important contribution that came from PET studies, approximately 1986, from Fox and Rakel. And what they showed is that oxygen consumption in the brain does not increase commensurately with blood flow. So yes, blood flow increases coupled to neuronal activity, but oxygen consumption does not go up as much. And when you have that particular property, then you have a situation where the paramagnetic deoxyhemoglobin concentration changes in the brain. So deoxyhemoglobin is in blood vessels, and in these blood vessels, for example. And when you have a paramagnetic molecule sequestered in these blood vessels, the magnetic field around these blood vessels become non-uniform. We want the magnetic field to be very uniform in imaging, but there's a non-uniformity that comes when there's deoxyhemoglobin, which is specifically, it has to be sequestered in the blood vessels. And it's this non-uniformity that changes. It's an incredibly small effect. We were really surprised that we could actually detect it. And it is this particular phenomena, in other words, coupling to the neuronal activity through so-called neurovascular coupling, that we are monitoring. Now, when we published that paper, we had also quite a lot of modeling and theoretical work on that. And we published a second paper, and where we actually looked at this particular effect mediated through the blood vessels. This is a picture of what blood vessels look like in the real brain. You have, large vessels up here on the surface of the brain. You have somewhat or medium-sized vessels going perpendicularly into the cortex, and then you have these capillaries. And our modeling showed us that the effect that we were monitoring comes from large and small vessels. And the large vessels, which we don't really want to see that much, they increase as linearly with the magnetic field. So B0 designates the magnetic field strength. And the signal that comes, okay, so the signal comes from the capillaries, increases as the square of the magnetic field. So this is essentially told us that, okay, magnetic fields are very important. And this is a somewhat more advanced modeling that came much later in 2009 by Kamil Uğurbil, who was working for me when I was at the Max Planck Institute in Tübingen. And essentially that shows the fMRI signals, you can see over here, depend very strongly on the magnetic field. These are two different kinds of fMRI signals. You don't have to know what they are exactly, but the most important part is that they increase supralinearly with the magnetic field. So we knew from the very beginning that magnetic fields are going to play a very important role. There is another thing that we have to be aware of, and namely, in fMRI, the field developed very quickly so that you can obtain maps like this. And these are approximately, say, 2 millimeter resolution. But of course, this is a very coarse scale for the brain. The brain operates over many different scales. For example, there are organizations, so-called columns and layers, overshown here. And these are so-called orientation domains. It's a very canonical organization that is studied a lot by electrophysiology, optical imaging, and now fMRI. And these are approximately less than 1 millimeter scale. And these are the population of cells that respond to different orientations. And then there's, of course, ocular dominance columns, one eye versus another eye. So this is so-called the mesoscopic scale. And then, of course, we have the microscopic scale of single neurons. Now, this cartoon that depicts this orientation domains, really, orientation domains were discovered by electrophysiology. So David Hubel and Torsten Wiesel, they were doing electrophysiology studies, sticking electrodes, in this case, into cat brains. For example, they're recording from a single neuron here. And in this case, this particular neuron, for example, responds not at all when the orientation is like this, but orientation like this, it responds, and like that, it doesn't. So there's some sort of a response, tuned orientation response. And the interesting thing is, they tried to find out how are these neurons organized with electrophysiology. They couldn't, or they were wrong, let's say. They did propose something, they were totally wrong. And how those neurons were organized were discovered by optical imaging. And this is about the time that fMRI came into existence. A paper like that also showed up. fMRI papers were published in 1992. This is in 1991. This is optical imaging of intrinsic signals. So they are looking at hemoglobin. So it's kind of like fMRI, but not quite, because they see total hemoglobin. We are sensitive to only deoxyhemoglobin. And they were all able to show that in the animal brain, these orientation columns are organized in this particular fashion with some singularities like this. And around the singularity, all orientations are represented. It's a very fascinating, I thought that this was really fascinating, because how the brain solves the problem of representing 360-degree orientation. And so it became very critical for me. I'm pausing every once in a while because the switching of the slides are not very smooth, but it was very critical for us, the question that we were asking, can we extend fMRI to actually cover these mesoscopic scale organizations? Then, but there were other questions when we asked that. There is another question that, these mesoscopic scale organization, I should say, not only involve these orientation, but also these cortical connections, which are actually based on these layers. And this is approximately a few millimeter thick cortex, so-called cortical layer. But when you ask that question, can you can you extend fMRI to cover that? There are really two particular questions. These same optical imaging people, Maybe I should stand over here a little bit so I can see what I'm talking about and point out. There were two, same optical imaging people actually also said, well, you know, when you look at optical imaging, you know, sensory stimulation of cortical columns initiates tissue hypoxia and vascular response that occurs within the first 3 seconds and highly localized to individual cortical columns. But if you wait for a while, then that accuracy disappears in 3 to 5 and becomes, they spread over 3 to 5 millimeters. And their concluding sentence was, this large mismatch between oxygen consumption and supply probably underscores the importance of adequate oxygen supply, watering the entire garden for the sake of one thirsty flower. Because we are in fMRI, we are looking at watering the neurons, then this sort of implied that fMRI may not be able to actually extend to that particular level. So I don't have time to describe it, but you know, we did a lot of experiments in animal models looking at blood perfusion, and we actually came up to the conclusion, this is published in this paper, you know, blood perfusion is regulated at the level of cortical columns. And okay, I have to go back here. And perfusion response has we measured the full, you know, full width half maximum, it's about 470 micrometers. And we came up with the conclusion that, well, you know, this idea that brain waters the entire garden for the sake of a thirsty flower is wrong. And instead, actually, brain waters the thirsty flower, but it generously sprinkles around it as well. So that was the physiological answer we had to get to give us the confidence, saying that we can actually maybe, you know, map activity at that level. So what else do we need then to get to that level of activity? Most importantly, we need signal to noise and contrast to noise. And let me tell you what I mean by contrast to noise. So for example, if you are looking at a functional imaging response, let's say that you are subject is sitting in the magnet, you turn on the lights and the signal changes, you turn off the light, the signal comes back, and there's some noise because the signal is fluctuating. So this particular effect relative to this noise, you can define that as contrast to noise. I don't have a lot of equations, but this is one of the few equations I will have. So you can actually recast that equation to contrast the noise. You can write it like that. This is just a simple mathematical expression, manipulation. And this N is thermal noise at high resolutions. I specify that because in fMRI, when you go to low resolutions, there are other noise confounds that come in, and you can write it like this. So we know that this here is the so-called Bold effect, and we understood this from modeling. As I had mentioned, it increases with the magnetic field somewhere between linearly to quadratically. And at the time, we didn't know exactly how image SNR would scale with magnetic field, but we were very happy to explore this and then find out how this went. And today I can tell you, in fact, just in a series of papers we just published recently, that also goes somewhere between linearly to quadratically with magnetic field. So you come up with something like functional imaging goes somewhere between quadratic to fourth power with magnetic field. Hence, magnetic field sizes are very important, and 7 Tesla is what we introduced initially to get to this level of cortical columns, did in fact, after many years, gave us these functional images in the human brain of ocular dominance column, one eye versus the other eye, and the orientation columns, as you can see over here. And this brought fMRI in the human brain, shown here for these columns, to the level of what was possible with animal models. In this monkey, for example, optical imaging. Of course, this resolution is not as good as this one, but this is an invasive technique. You have to put a hole in the brain, you have to, on the skull, and expose a very little area, and you cannot penetrate more than that, whereas this we can do in the entire brain. And I mentioned layers, just to show you that this is also now very, not easily, but doable, thanks to very high magnetic fields. In this case, I'm showing you again visual stimulation, and visual stimulation input into the brain comes into middle cortical layers. And you can see it here coming into middle cortical layers and easily detected. So today, 7 Tesla is becoming very popular. It's commercially available. Our first 7 Tesla was, of course, not a commercial instrument, but it's now commercially available. And when you look at now a growing number of high-resolution layer fMRI studies, all being conducted at 7 Tesla, but very small amount of work being conducted at 3 Tesla and some at 9.4 Tesla. So 7 Tesla, it's increasing. We don't have one in Turkey yet, but it's North America, Europe, and Israel has 7 Tesla. But 3 Tesla is all over the place, but unfortunately, it doesn't have the power to get into these very high-resolution MR studies. So, but high-resolution MR is not the only thing that we study. We actually do other things with fMRI. So one of the things, this is coming from the Human Connectome project, and namely, instead of looking at stimulus or task with fMRI, you can just look at the brain when the person is just sitting in the magnet and doing nothing. So we call this resting state fMRI. And if you look at the signals, they are, as you can see, oscillating. But those oscillations, these are for you, your request. So those oscillations are not random. And in fact, you can see that they are correlated. There are 3 different regions and 3 different colors. They are correlated between those regions. And if you actually map that correlation, so correlate spatial patterns of correlated activity, you get something like this. That looks like a network in the brain. And in fact, you can generate what we call a connectome matrix. So you can look at those oscillations, and you can correlate every single voxel in the image with every other voxel in the image. And this is a matrix that represents your brain. It turns out it's very unique to your brain. We can even identify individuals. We can, differentiate between individuals based on this connectomics mix. It's like a fingerprint. Now, you can look at this in this matrix form, but if you were to say, what is this single line, for example, these 2 single lines in the matrix, they correspond to images like this. For example, you have a seed, and you look at correlation of this particular point with every other point, and you can look at this particular case, and you get a connection. These are, in fact, it turns out, they are networks in the brain. You can really, if you know these correlations, you can even tell how that particular brain will respond to a task or a stimulus. Now, there's another methodology in the Human Connectome project, so-called diffusion imaging. I don't have the time to go into it again, but we look at diffusion, and in the brain, diffusion is anisotropic because of all the connections in the brain and these fibers. Water can diffuse along axons, but it cannot cross them very easily. And from that diffusion imaging, you can actually get connections in the brain like this. These are actually representations of connections. And in the Human Connectome project, we can generate data like that. I don't know if you can see that video. It's actually a little bit too bright, but you can generate data like that, where the diffusion imaging, in this case, is looking at arcuate fasciculus, which is a connection in the brain like this that connects language areas. And you can also get the language areas from these spontaneous oscillations, and you can correlate them together. So this kind of is a study that was funded, and it continues with Human Connectome project. And the questions are now, this connectomics makers and this connectivity, whether it is functional or anatomical, is it changing with development? Is it what's happening with aging? What's happening with different diseases? So it's a very ongoing and exciting project that involves fMRI. But let me come back to ultra high fields again. So I mentioned to you that we were able to go from here to over here using fMRI. And of course, now we are very much interested, oops, sorry, the wrong way. pushing this resolution even further towards microscopic scale. And there are many reasons for doing that. One of them is, in fact, this, I should tell you, this is an optical image. It's a 2-photon image of activity. So these are, each one of them is a single neuron, cell body of a neuron, and you can see a little bit the axons. And you can, with optical imaging, you can detect, when you detect these neurons, you can actually monitor the activity. And multi-photon optical imaging, the scale goes from signal neuron to something like this, that these two scales can overlap. So we are very much interested in this overlap because MRI, after all, has a limitation somewhere over here, and optical imaging has a limitation also somewhere over here. They cannot go this way. fMRI does not go this way. So we are pushing multimodality, and these are the people from CMRR in collaboration with Purdue University investigators developing optical imaging inside the magnet so that we can do simultaneously fMRI and optical imaging. So these images are 2-photon images, and many of you may have seen such images. So in this case, there's a dye that is injected into the blood vessels, and you can see the blood vessels. And here you are spanning depth. You go from the surface, you have the big vessels, then you go in, then you only see capillaries. And in this case, you are just focusing on a single plane. Yeah, you're looking at it as a function of time. You can see these dots going back and forth, I mean, not back in one direction. Those are red blood cells, individual red blood cells. And you are looking at individual red blood cells flowing in the capillaries. Now, this is a vascular image, if you like, 2-photon. But the unique thing is that it was obtained within a 16.4 Tesla magnet. And if you look at what kind of fMRI resolutions we can get, it's something like this. This is, in this magnet, we can get an fMRI resolution of 250 micrometer isotropic. It corresponds to a scale like this. So from the point of view of modeling the functional imaging signals, being able to do this vasculature on a voxel by voxel like that is extremely important. But of course, what is much more exciting is the fact that you can at the same time not only look at vasculature, but neuronal activity as well. So this is, for example, the rat, this in this case is a mouse brain. There's a plastic clear window here, and you can obtain various different kinds of images. But an image like this right over here now, you can also obtain with optical imaging, looking at calcium signals as shown over here. These are, each one of them is a neuron now simultaneously with fMRI. There are still a lot of neurons there, but they are it's a tractable problem, so that you can actually look at every single neuron. And here you are seeing the spontaneous activity of these neurons, but these are not fMRI signals. These are actually neurons firing. They are spikes, if you like, but monitored with optical imaging. And we can then correlate it with fMRI signals and look at analysis and trying to understand how these neuronal activity actually enters into fMRI. So the idea is that in this particular setup, we can go from a single neuron to whole brain networks, but using two techniques, optical imaging and fMRI. Now, I want to backtrack a little bit. I emphasized that we were focused and still focused on tool development, and this is an example of that. But let me come back to magnetic fields again. So we pushed magnetic fields, and I want to just sort of impress upon you that it wasn't like you buy a magnet and you connect it and everything just falls out. There was quite a bit of skepticism about high magnetic field and human imaging, because it's actually a bit of a problem. And this is a paper by two very well-noted investigators, David Holt and Paul Lauterbur, who did a modeling study in 1978, published in 1978. And their conclusion was, well, effects associated with radio frequency penetration depth suggests that the frequency of operation of the spectrometer with human samples should be less than about 10 megahertz. That's the RF frequency. That translates into 0.23 megahertz. So there was quite a bit of skepticism, as I mentioned, that physics would allow you to do ultra-high magnetic fields. Now you can say, well, given this very well-known investigators, Paul Lauterbur, in fact, won the Nobel Prize, why did you go up to very high magnetic fields? And luckily, I had not read this paper. And as a result, I was ignorant, if you like. And in this particular case, ignorant was bliss. But even if I had read it, I would have still done the experiments, because this is a theoretical study. Theory is extremely important, but has to be experimentally verified. And in fact, there was quite a lot of other data when 4 Tesla came into existence. Actually, 4 Tesla we did not start. 7 Tesla we started. 4 Tesla companies started it. They produced MR images like this when they got 4 Tesla. And at the time, they could actually obtain much, much better images using 1.5 Tesla. So there's a problem at 4 Tesla. And they actually more or less abandoned the field of pursuing 4 Tesla. And the problem comes from the fact that wavelengths of the RF that you are using becomes small when you go to high magnetic fields, and they become smaller than the object that you are trying to image. For example, for the 7 Tesla, human head is approximately 20 centimeters, let's say, and the wavelength of the RF we are using is 12 centimeters. And if you want to look at human torso, it's even worse. Now, wavelength is 12 centimeters, but we are talking about 40 centimeters and larger. So this is a new regime for MR imaging, whereas at 1.5 Tesla, it's just the other way around. So when you go to high magnetic fields, we are operating in the so-called far field regime, as opposed to the near field regime, which MR experiments operate at that time. And luckily, we can actually do simulations, electromagnetic simulations, and look at what happens in this particular far field regime. So this is 7 Tesla simulation. Here's an RF coil. It's a very relatively small size compared to the human body. And whereas at 1.5 Tesla, everything would just light up and disappear when the RF is being transmitted. And you can see here that it's actually a wave behavior. Actually, the B1 field has to be generated. It penetrates the human body slowly, and that particular penetration, it actually even goes to areas of the human body where this RF coil doesn't cover. This does not happen at low fields. So we had to overcome this with the technology, and the technology we introduced was essentially so-called multi-channel transmit. So instead of using a RF coil, which is a single channel, we do multiple channels, and we transmit RF with multiple channels, and we receive them with multiple channels. And this is a technology now that is common and used in even commercial machines. But this is some of our recent experiments, recent developments. So these are quite a lot of engineering development about these detectors: multi-channel transmitter, multi-channel receiver for head imaging. These are multi-channel transmitter and receiver for body imaging. So with this technology, so when 4 Tesla imaging looked like this, we were able to go to images like that at 7 Tesla and in the brain. And for human body, when 4 Tesla images generated, in this case by Siemens, looked like that, and in 2009, we were able to demonstrate that you can do imaging in the human torso. Now, of course, I told you we developed high magnetic fields because of functional imaging, but we are very sensitive to what other applications that we can do. And images like this, in fact, was part of a paper looking at the diagnostic quality of those images. So you are looking at the hippocampus. Unfortunately, this bright light doesn't allow you to see this image very, very well. But here's the hippocampal structure beautifully delineated in the human brain. And we did this because actually a neurologist colleague came to us saying, okay, I have a patient, I think it's temporal lobe epilepsy, but I don't see it in the 3 Tesla images. Can you help me? And the result were those images and these images also. Here's the control subject, beautiful hippocampal formation. And these are 3 patients where you can see hippocampal is actually, hippocampus formation is very different. And these are temporal epilepsy patients. So what we did for fMRI, in fact, also helped the clinics, and 7 Tesla today is maybe the most advanced clinical MR system for brain diseases. I want to come a little bit, I guess already I did quite a bit of technique, but I want to come back a little bit again to our engineering efforts, just to give you a flavor. I mean, some of you may not be, depending on your background, may not be interested or able to follow this, but I want to give you the flavor that, in fact, engineering is a very important part of our effort. So I mentioned to you that signal to noise is very important, and we have to increase signal to noise. So we go to higher magnetic fields. But does the signal to noise increase at high magnetic fields? This has been a major issue, or how does it increase? So for example, this is a paper we published recently in collaboration with in Maastricht University, who has a 9.4 Tesla. 10.5 Tesla is what we have. We all have 7 Tesla and 3 Tesla instruments. And France, CEA, recently achieved 11.7 Tesla as well. So the 3 groups collaborated. We had a phantom like that. We shipped from place to place. It's a spherical phantom that has electrical properties that actually mimics average electrical properties of the human brain. And you look at these images, 3, 7, 9, 4, 10.5 Tesla, 11.7 Tesla. And the blue color means the signal starts high here, it actually decreases as a function of the magnetic field. But only in the center, it looks like it's going up. So if you were to just look at this data, you would say signal to noise does not necessarily increase as a function of magnetic field. But we actually knew already that this would be the case because of a paper we published in 2005 that showing that this is a particular property comes from the particular geometry of the shape and the particular geometry of the RF coil. So we knew that you cannot talk about signal to noise increase with magnetic fields over here. This is because there's destructive interferences, but we can talk about signal to noise increases with magnetic field in the very center of this phantom. And indeed, in the center of the phantom, you can see that magnetic field increases and it goes out square as a square of the magnetic field. Very satisfactory for us. And we also did calculations, again, very good agreement with the experimental data. But this is also in the center. So how do we actually talk about magnetic field increases outside of the center? Now, a lot of that comes from modeling. We launched a modeling effort comparing 7 Tesla signal to noise to 10.5 Tesla signal to noise. And we are asking the question now, independent of any RF coil, theoretically, what is the maximum signal you can get in the human brain? And it turns out something like this. You can see that signal noise is ratio 7 Tesla to 10.5 Tesla is high in the center, but somewhat linear in the periphery. So in the center, signal noise goes up, something like 2.5, factor of 2.5, going from 7 to 10.5 Tesla. And the periphery, it goes approximately, at the edge of the brain, approximately quadratic, at the edge of the head, linearly. So the question then, a physics and an engineering question, how do we capture all of the signal noise, not just the center, but edge of the brain? I mean, that's extremely important. And of course, we develop coils like this, sort of following a pathway that exists even at 3 Tesla, multi-channel receivers. And we look at, these are phantoms that are like human brain, human head shape. You go from 7 Tesla to 10.5 Tesla, you get these gains. And when we look at those gains as a linear plot, what we did is we took these sort of shells, and because the problem is somewhat spherically symmetric, we are looking at the average SNR in these shells going from the periphery to the center, and we plotted them for two different kinds of arrays. Essentially, we thought, okay, if we build the same array for 7 Tesla at 10.5 Tesla, we compare it, we should be able to understand the signal-to-noise gains and capture it. And it turns out, yes, we meet theory on the edge, on the periphery, but in the center, we are missing it quite a bit. So the technology that you have at 7 Tesla doesn't quite work at 10.5 Tesla. And we had to go back to the drawing board, essentially, do a lot of simulations and experiments. Essentially, what turns out, the technology at 7 Tesla, which captures, let's say, theoretically, let's say about 90% of the theoretically maximum SNR, fails to do so at the 10.5 Tesla. These are same coils. You go to 63 channels, it's the same thing, and this is experiment. Even though the values are different, the trend is the same. So we had to think about what we have to do, and essentially we are trying to detect current patterns on this particular shell, and they are getting more and more complex when you go to high fields. And how you can capture that, one approach is to have many channels, so that all those many channels can capture a complex current variation, in a more accurate way. So this is a 128 channel coil. It's, as you can see, it's rather complex. It's completely developed at home. We had to miniaturize preamplifiers, and we have to also minimize interactions between a transmitter and the preamplifier. I'm going a little bit into engineering detail only to show you that there is quite a lot of engineering effort. And then once we did all of that, in fact, now 128 channel at 10.5 Tesla looks like what 64 channel can do at 7 Tesla. And now we can talk about here, now experimentally, are we really getting the theoretically available signal noise gains? And indeed we are. We can go more than square in the center here, right over here, 128 channels, and approximately linear. So there's quite a lot of engineering effort, as I had mentioned, and but it's not just engineering. We have to get signal to noise from the instrument, but we also have to, there are ways of getting signal to noise from image reconstruction. I will show you two examples. One of them is this effort, so-called Nordic, and essentially this is not a deep learning method. It's actually based on low rank properties, and this is why the mathematics comes into place. So these are functional images of two different stimulations in the visual cortex represented on a flat brain. And you have 5 minutes of data, 15 minutes of data, 25 minutes of data, and you can see that you are signal averaging, and of course your images get better and better, and you can actually, about 25 minutes will give you a good depiction of these visual areas. But using this low rank properties, we can actually reprocess the one single run, 5 minutes of data, and get exactly something that looks like this. So this is a paper that was published in 2021, which is now used quite often to improve signal to noise just by image reconstruction. And of course, today, deep learning is a very big area, and it comes into imaging as well. And this is just an example from deep learning reconstruction. So this is just conventional linear methods, activation in the brain with visual stimulation, and the color maps are activity in the brain. Background is anatomy, and this is half the data, but now this is physics-driven deep learning, and half the data you can get actually by these deep learning methods, something that actually looks like what you had with the full set of data. I have to say, we more or less pioneered this effort, and it comes from the lab of Mehmet Akçakaya in electrical engineering at University of Minnesota. And the very first paper we published in this area actually won a prize in 2022. A paper was published in 2019. 2022, in this big meeting, it was received the first prize for the most highly cited paper. And the technique is called scan-specific robust artificial neural networks for case space interpolation, RACA, in other words. So this is the RACA technique based on deep learning and gives you a lot of advantages. Let me just end by, on a lighter note, maybe easier, just showing you some images that we can obtain with all this development that we are getting. I won't show you any deep learning images or denoised images. Well, actually, I'll show you some denoised functional images. But these are some anatomical images now from 10.5 Tesla. And this is the human visual cortex, cerebellum, and you can see the cerebellum now very beautifully depicted there. And here, this particular image was really exciting for me, because if you know visual cortex, there's something called calcarine fissure. And this is essentially where the two banks of the primary visual cortex is separated. So this lower part and upper part of this calcarine fissure is so-called primary visual cortex, or V1. And V1, you can, of course, identify functionally, but you can also identify, this is one of the unique areas, you can identify anatomically. It has a marker, so-called line of Generi. Line of Generi is a bundle of myelinated axons that run in the middle of the gray matter, which is very unusual. This is the only place in the brain it happens. And you can see the line of Generi very beautifully in these images. This is the dark line in the middle of the cortex. Let's see if I can. And you see it in the upper V1 and the lower V1. I mean, there have been images in the past that show that those line of Generi maybe can be visualized, but so critical, so beautifully, and both on the upper and lower parts, banks of the V1, I've never seen before. This is another image on the same plane. This is obtained now with this 128 channel coil. Again, very beautiful depiction of the cerebellum. And anatomical images somehow, I think, has a visceral appeal to people. So they made covers. So this one I showed you made magnetic resonance in medicine, the cover. And this particular image was in the news in Nature Methods, showing, pushing the limits of fMRI brain imaging. Okay, some more images. This is an axial slice from 10.5 Tesla. A lot of structure here. I mean, there's also a lot of resolution. We can expand it. And for example, you can see a lot of the intracortical blood vessels, connections over here. So quite a lot of anatomy. And MRI, of course, you can use many different kinds of contrast, right? You can do functional imaging, you can do anatomical imaging, different kinds of anatomical imaging. This is so-called susceptibility-weighted imaging, which is sort of like functional imaging in the sense that it is sensitive to areas of the brain which contains paramagnetic deoxyhemoglobin, namely veins. So you see the veins, these are 3 slices, beautiful depiction of the veins in the human brain. fMRI at 10.5 Tesla. This is auditory cortex stimulation. These are whole brain studies. I am emphasizing the whole brain studies because I told you it is a really, it was a challenge to capture the SNR at 10.5 Tesla in the whole brain. And these are the highest resolution fMRI studies that now exist. It's approximately 0.6 millimeter isotropic. But it has, just to demonstrate the power of the magnetic field games, it has no denoising and no smoothing. And our record so far is 0.35 millimeter isotropic functional images in the visual cortex. I will end by just simply telling you, without going into detail, but okay, we work on the brain. This is our main activity, but not only. We also focus on imaging other organ systems. They are much more clinically oriented efforts. So this is effort led by Greg Metzger, showing that you can actually do imaging of the torso in 7 Tesla and with higher signal to noise. You can even do it at 10.5 Tesla. And for a change, we are not only just working at very high magnetic fields, but we're also working at somewhat lower magnetic fields. So this is a new concept led by Michael Garwood. And the unique thing here is the magnet, as opposed to those big structures that you see, is this little gray thing. So you sit on the chair goes up, your head goes in here, you can look out the magnet, for example, talk to your friend or your partner here while we actually image your brain. Okay, look at the activity of your brain or the anatomy. So this is also a brain initiative development that is taking place. And the unique thing here is not only this magnet, it actually was meant to be 1.5 Tesla, but often, as happens when you're pushing the technology, it didn't quite make it to 1.5. It's at 1.7, but we understand why it didn't make it. It's just a matter of quality of the wire we have to get, because the wire is high temperature superconductor, unlike the other magnets. So the wire quality is difficult for magnets, but the magnet is very small. That means the magnetic field is very non-uniform. So now you have to develop techniques for imaging in non-uniform magnetic fields. So it has its challenges and also rewards as well. With that, I want to end, again, coming back just to acknowledge the fact that a lot of people, of course, contributed to all this work. And some of them, you've seen their names mentioned. And there are many others who are not even in this picture, contributed to the work. And now they have their own centers and own labs. And I'd like to thank you for your attention.
Would you like to take some questions?
Of course, I'm delighted to take some questions.
So you need to get your microphone. Not here. Get back. Here first, that goes. We'll stop. And then.
Thanks for the great talk, Karen. My first question is, would you ever yourself go inside a 10.5 Tesla MR to get your brain imaged? In that sense, how is the safety concerns for live humans in the ultra high fields?
You know, I hate to say this, but I have not been into 10.5 Tesla. And okay, I will tell you why. The reason is we do operate under FDA. We have under FDA investigational device exemption. They allowed us to put a limited number of subjects within a particular age range, and I am not in that age range.
But it's okay for the other.
But we have put many people. You've seen 10.5 Tesla images, and those are actually we do. It is right now quite a cumbersome procedure to do imaging at 10.5 Tesla, because we have to do something like 1 day of testing before they go in. We test, and then they come out. We test, and so far everything is okay. We are reporting it to the FDA. We look at physiological parameters, we look at cognitive function, etc. And so everything looks okay. So we are pursuing 10.5 Tesla. And French group, I mentioned that they went to 11.7 Tesla just recently. They've been working on it for quite a long time, but they recently achieved 11.7 Tesla. And they did report preliminary human images, so they put some human, but they don't have such a large number of people so far. We have quite a lot of people. It's something like, I don't remember, but maybe like 50 people, 160 exposures or something like that.
That's great. So also some technical questions. So in the live imaging, the human breathing and many things, when you try to go into very highest resolution, that you show beautiful images. Can that work while we have these in vivo activities? Are they, or do you need to correct such motions for the highest resolution? And also, what's the importance of gradients that you didn't mention? So because you showed the B0, which is very important, but what about magnetic gradient magnitudes? That's how important are they for the high resolution imaging?
It's very important. It's actually there is two confounds that come in MR. There are two confounds. One is some areas of the brain do pulsate, especially in the center of the brain. Visual cortex not so much, but other areas move. And the other thing which is unique to MR, so when you are breathing, your lung volume changes. So air has different magnetic susceptibility than tissue. So the magnetic susceptibility of this large volume near your head is going up and down. So that means that it actually perturbs the magnetic field over your head. So the magnetic field is fluctuating over your head, and you have to correct for that. So we put a lot of effort actually in correction, and there are methods that are based just simply on the NMR acquisition. You can so-called do navigators-based techniques, which we are working on a lot. But there are also other methods. You know, your colleague Klaus Prusman, who actually started that when he was in CMRR, you can put these probes around the head, and from those probes you can measure the magnetic field and try to estimate how the field is changing over the brain. So you have to do, indeed, you have to do corrections. And of course, when you do optical imaging, it's the same thing you have to worry about. There, it's only motion. It's also through, it's only through plane motion that you have to worry about, and you have to correct. It's a very big area of research.
Thanks.
So it's great talk. I really enjoyed. So my question is that if I got that right, you said that fMRI result is signature is unique from person to person, right?
Yes.
So how is that possible? I mean, you're looking at the neuronal cell, and why, or what is the source of that uniqueness you think in getting from different from one person to another? Because
Well,
look at the composition, brain composition. You have water, you have organic molecular matter, basically. Why do you think you're getting a different signal?
it's unique to the brain not because of brain composition, but because of neuronal activity, right? So you are actually you are making a measurement about the spontaneous neuronal activity in the brain, and that's unique to people. Now, why that is unique to people, we can speculate on that, and you know, it's essentially it may reflect the, you know. It may reflect genetics, and in fact, expression of some of the enzymes have been shown to also correlate with those spontaneous activities. It may also represent experiences and the networks that have evolved in your brain as a result of your experiences.
Thank you.
And of course, we are also hypothesizing, I should say, that those networks are going to be different in different disease states. And there are a lot of people who look at those, study those networks as a function of disease. And they've been shown to differ for different disease states.
There's a question all the way on the top. Had her hand up. Can you talk to the microphone? My voice is looks loud, but you can hear me.
Here's you have the microphone has reached you.
So considering that Earth's gravitational field is approximately 50 microtesla, this is a beast of a machine that has a very strong pull. The thing about MRI and fMRI is that, like, MRI is 3D, fMRI is 4D. There is temporal dimension as well. And when we collect data, as the time progresses, the head goes more and more into the blanket, and there is unavoidable head motion. And the thing is, if the voxel size is relatively large, the content of the voxel doesn't change from head motion to head motion, but if the voxel is very small, then even the smallest head motion can change the whole content very drastically. My question is the head motion another important variable that you deal with differently as opposed to smaller fields? After all, we are not measuring dead salmons. We are measuring like live humans.
Yes, I mean, this is very much related to the question that Metin was asking. And I have to say, when I was answering Metin, I was talking about brain pulsation and these fluctuations, but this is another kind of motion. In other words, rigid body motion of the brain, not the brain or the head, actually, it's rigid body motion. You gave the example of the head sinking into the, cushions that you have, but the subjects can be also, moving the head, etc. So, and you are very right. And when you are working at very low resolution, that motion may be small compared to your, voxel sizes, and they're not so dramatic. You can correct it by post-acquisition methods. But yes, when you go to much higher resolutions, you really have to pay much more attention to motion. Otherwise, you will not be able to register one image after another, because fMRI, as you point out, is a time series. So we try to take care of both prospective and retrospective motion correction when we go to very high resolutions. So I should just add just rigid body correction. If you have good anatomical images, just retrospectively you can do a pretty good job, but only for rigid body. Yeah.
So thank you, Hocam, for the talk. I have a question about the size of the devices that you work with. So I understand that a lot of people in the field is working towards improving the field strength, like just improving the brute force. But the way, as an outsider, I see is maybe perhaps for wider adoption, smaller devices would also help a lot. How do you see, like in the future, if we could utilize, let's say, 7 Tesla or 10 Tesla devices at a very small size that maybe tens of them could be available in a hospital?
Yeah, of course, miniaturization is important. The problem is being able to build a 10 Tesla device or a 7 Tesla device that is very small. And that's a major challenge. However, there are magnet companies that are working on that. I showed you an effort by us, but not 7 Tesla, 10.5 Tesla. It's 1.5 Tesla at the present time. But in principle, that can go to higher magnetic fields. And unfortunately, when you make a magnet and you want to reach a high magnetic field, there are maybe, if you like, 2 ways of getting there. One of them is you pass more current through the existing wire, and so to achieve a field, and that has challenges because of the fact that, with more current, local magnetic fields are much higher, forces are much higher, you have to contain that. And also the wires cannot necessarily tolerate a lot of current. At some point, they have a limitation in terms of current carrying capability and remain superconductive. So there are challenges there. The other way you can actually achieve high magnetic fields, but you can keep the current low, but you put much more wire, and that means you get a big magnet. So there are engineering challenges if you want to say, okay, I want a small magnet, but at the same time high magnetic field. But of course, yeah, I mean, this is mainly, I think it depends on wire technology, and I think that high temperature superconductors are very promising in this respect, and but their manufacturing is still not so good. They are increasing as demand increases right now, and I think the wire quality will get better. Luckily, I mean, building magnets is not commercially and sufficient to drive wire production. On the other hand, there are many other, not many other, there are some projects which do require a lot of high temperature superconductors, and they are driving the manufacturing of this wire and making it commercially feasible. And particularly fusion now is a big topic now. And right now, there are several producers of high temperature superconductors in the United States. You cannot buy wire from them. They're all committed to fusion, already sold, if you like. So I think future looks pretty good. Maybe we will have much smaller magnets.
Maybe just briefly, so when you work with higher Tesla, you build the magnets yourself or you purchase them? For example, this 10.5 Tesla.
So we can deal with, there's quite a lot of electronics, and I showed you some of the detectors, and there's, of course, digitizers, this and that. We can deal with all of that, but we really don't have the ability to build a magnet like that. So these are, very few companies can build them. And so they are manufactured. And we were lucky enough to partner with a very small magnet company that was trying to actually get into the business of manufacturing magnets. And they were willing to work with us to build these magnets. Well, although this magnet, this one it was developed as a research project in Siemens laboratories.
Thank you.
Thank you for speech. I want to ask about magnetic stimulation, TMS, transcranial magnetic stimulation on some patients, like increasing neuroplasticity or neural activity in obsessive compulsive disorder or depressive symptoms. And they check using the fMRI changes. And what do you think about this usage of rTMS or TMS in these disorders? What is the changes of the brain in this usage?
What do I think about it? Well, as you mentioned, TMS is being pursued in terms of as a means of doing external stimulation of the brain. And I know that there are potentially some success stories, but it has not become a mainline, a treatment, so to speak. But people are trying, and of course, it's also used as a research tool. For example, you can stimulate the brain locally and look at, if you were to stimulate the brain locally, where else are you getting activation? So that's one way of looking at connectivity in the brain. And so it's combined. It's doable together with fMRI. It's somewhat difficult because if you have a TMS, which in itself is like a little pulse magnet, so there are quite a lot of forces on it in the magnet, inside the magnet, but it's doable. Some people are interested in doing that. We are not pursuing it ourselves so much, but it is, yes, it is. It's an experimental tool, if you like, in addition to fMRI to study the brain.
Thank you very much.